Hello, everyone. Thank you all for joining us today to hear about incorporating spatial mapping into your single-cell biology workflows with the RNAscope technology. My name is Courtney Anderson, and I am the Research Manager of Applications at ACD, a Bio-Techne brand. Complex, highly heterogeneous tissues, such as the brain and tumors, as well as processes such as embryonic development, are comprised of multiple cell types and states. Precise transcriptomic characterization of these tissues can enable identification of new cell types, predictive biomarkers, new therapeutic strategies, and more. However, interrogation of these complex heterogeneous tissues requires a highly sensitive, specific, and multiplex spatial approach with single-cell resolution. Traditional transcriptomic analyses have relied on bulk tissue analyses, such as PCR, which provide an average level of gene expression in a sample but loses the spatial and single-cell information. This is akin to a fruit smoothie, which can be a delicious drink, but you can only guess at what is in the smoothie. The advent of single-cell transcriptomic analyses, such as single-cell RNA sequencing, have now been able to provide molecular information at the single-cell level, and to come back to the fruit analogy, can now provide you the menu and tell you what fruit and how much of each fruit went into that smoothie. However, these techniques still don't provide you with how those cells are organized in the tissue context. And this is where spatial analyses come into play. Single-cell multiplexing spatial analyses that detect RNA can provide transcriptomic information with spatial organization while retaining single-cell resolution. So now instead of an average blended smoothie with mystery ingredients, you can see all the fruit and how it is spatially arranged. The RNAscope technology is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single-cell level. It consists of three parts, which I'll describe in more detail on the next slide. First is a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots. The assay allows for the spatial mapping of messenger RNA, long non-coding RNA, splice variants, and highly homologous sequences in cells and intact tissues, all of which can be visualized with either fluorescent or chromogenic labels. And the assay can be performed on a wide variety of sample types, including FFPE tissues, fresh frozen or fixed frozen tissues, PBMCs, and cultured cells. Now again, the two key features of the RNAscope technology are its probe design and signal amplification. The oligonucleotide target-specific probes are depicted as Zs to emphasize the fact that they have two regions linked by a spacer. The bottom of the Z complements and hybridizes to the target transcript, while the top of the Z is the base for the amplification structure. When two Zs hybridize in tandem to the target sequence, it creates a binding site on which a preamplifier can bind, and the amplification tree can then be built. Now, after the Z pairs hybridize with the target RNA, the preamplifier binds to the top of the ZZ pair. Each preamplifier can bind multiple amplifiers, and then each amplifier can further bind multiple labeled probes. These labeled probes contain either a chromogenic enzyme or a fluorophore. This signal amplification strategy yields high sensitivity and allows for visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule that can be quantified. Background is eliminated because the signal is dependent on two Zs binding next to each other on the target sequence. If both Zs do not bind next to each other, then the preamplifier cannot form a stable hybridization and the amplification tree does not get built. Consequently, no amplification of nonspecific hybridization occurs, generating little to no background signal. Now, a standard RNAscope probe will consist of 20 Z pairs pooled together that are designed to hybridize next to each other along a target region, allowing for a tremendous amount of amplification and signal potential. However, a minimum of only 3 Z pairs is needed to bind to the target RNA sequence in order to generate enough signal for molecular detection. Taken together, this combination of probe design and signal amplification ensures a high signal-to-noise ratio. The RNAscope technology consists of 2 unique assays to detect RNA in the tissue context at single molecule sensitivity with single-cell resolution. The RNAscope assay is applicable for messenger RNA or non-coding RNA species that are greater than 300 nucleotides in length, which is the majority of transcripts. The assay is available on automated and manual platforms and can be selected as singleplex or duplex chromogenic or as a multiplex fluorescent assay. Our current multiplex fluorescent assay can detect up to 4 targets simultaneously, but we will soon be releasing our high-plex assay, which can detect up to 12 targets simultaneously in tissue sections. The BaseScope assay is a more specialized assay designed for the detection of exon junctions, splice variants, highly homologous or short sequences that are between approximately 50 and 300 nucleotides. We can also detect validated point mutations such as KRAS and BRAF mutations. BaseScope is a 1Z or pair probe or slightly more, and we have an additional amplification step that allows us now to get down to the single ZZ pair. BaseScope is available as a singleplex or duplex chromogenic assay on either manual or automated platforms. And shown here is a brief overview of the current RNAscope product line. As I just mentioned, we offer chromogenic and multiplex fluorescent assays. We also offer fully automated assays that are compatible with both the Leica Biosystems BondRx and Ventana Discovery instrumentation. The multiplex fluorescent assays have been the most widely used for single-cell RNA sequencing confirmation, as it allows for the simultaneous detection of up to 4 targets in the same cell and can be combined with immunofluorescence for the detection of additional protein targets. Now, the RNAscope technology has been widely applied for numerous technical and research applications. But today, I'd like to focus on the confirmation and spatial mapping of high-throughput data, particularly single-cell RNA sequencing data using the RNAscope technology. So current single-cell transcriptomic studies utilize dissociated cells and result in a loss of spatial organization of the cell population in the tissue context. Validation and spatial mapping of single-cell analyses can be obtained using assays that retain spatial organization, such as RNA in situ hybridization. The multiplexing capabilities of the RNAscope technology, coupled with its high signal-to-noise ratio, enable single-cell and single-molecule detection of transcripts in the tissue context, providing confirmation of high-throughput single-cell transcriptomic results, as well as adding spatial information to these data sets. Applications of incorporating the RNAscope technology into single-cell sequencing workflows include spatially mapping a cell atlas, visualizing gene signatures of newly identified cell subtypes, classifying highly heterogeneous cell types, confirming new therapeutic cell types, characterizing the immune landscape in the tumor microenvironment, identifying new potential immunotherapy targets, analyzing or predicting responses to drug treatments, and also confirming publicly available data sets such as the TCGA and tabula muris. I'd first like to show some examples of how RNAscope has been used to complement cell atlases generated by single-cell RNA sequencing. Last year, researchers from Sten Linerson's lab at the Karolinska Institute created a molecular survey of the mouse nervous system. They analyzed 19 regions from the central nervous system, peripheral nervous system, and enteric nervous system by single-cell RNA sequencing, totaling more than 500,000 cells, and they created a taxonomy of all identified cell types, shown here on the bottom right. RNAscope was used to confirm the identity and spatial diversity of many cell types identified in this study. Here I'm showing the distribution of astrocyte cell types. RNAscope confirmed the distribution of the neurotransmitters SLC6A9, which was abundant in the cerebellum, and SLC6A11, which was abundant in the olfactory bulb. In addition, ISLR and GDF10 mark the local astrocytes enriched in the olfactory bulb and cerebellum. And last and most strikingly, detection of MFGE8 and AGT in the tissue context by RNAscope revealed a distinct border separating the telencephalon, or front of the brain, from the diencephalon, or back of the brain. In the same issue of Cell, researchers from Stephen McCarroll's lab at Harvard sequenced almost 700,000 cells from the adult mouse brain to identify 565 transcriptionally distinct groups of cells and create an adult mouse brain cell atlas, which can serve as a reference for development, disease, and evolution. RNAscope was used to confirm several of these results, including the correlation of parvalbumin, SYT2, and NEFM among parvalbumin-positive neurons. And just a few weeks ago, this publication came out in Cell Reports that created an atlas of vagal sensory neurons in the mouse. Vagal sensory neurons are necessary for the control of organ functions such as cardiopulmonary and gastrointestinal systems, but knowledge on the complexity of neuron types involved is missing. Researchers from Patrick Ernfor's lab at the Karolinska Institute presented a comprehensive identification, classification, and validation of the neuron types in the jugular and nodose-derived vagal ganglia. 25 RNAscope probes were used to not only validate neuronal types identified, but also to quantify the relative proportions of these neurons. Here we show some representative data interrogating the nodose-vagal neuron. And just last week, the Human Cell Atlas, co-led by Sarah Teichmann, published this seminal paper in Nature Medicine describing a cell atlas of the human healthy lung as well as the asthmatic lung. With regards to asthma, using more than 25,000 cells from healthy control and asthmatic lungs, they revealed mucus cell hyperplasia in the asthmatic lung. The multiplexing capabilities of the RNAscope Fluorescent assay allowed for confirmation of a strong increase in mucus-ciliated cells in the asthmatic lung by detecting co-expression of MUC5AC, a secretory marker, and FOXJ1, a ciliated cell marker, in the same cells as indicated by the white arrows in the image on the bottom right. These cell atlas initiatives in the normal and disease states have now empowered researchers to combine single-cell analyses with spatial analyses to identify potential therapies for many diseases. Researchers in the field of hearing have long hypothesized that a diversity of neuronal cell types exists within the cochlea, and that hearing-related disorders are associated with selective damage of these cell types. But until now, knowledge of how cochlear neurons differ at the molecular level has proved elusive, which has hampered the exploration of novel drug targets for hearing disorders. Now, just to give a little bit of background, the inner ear contains the cochlea, which is responsible for detecting sound. The cochlea is lined with sensory cells called hair cells that convert sound vibrations into bioelectrical signals. These signals get processed and then relayed to the brain by neurons known as spiral ganglion neurons, or SGNs. And damage to these neurons can lead to hearing loss. These SGNs fall into two broad categories: type 1, which is the predominant cell type and responsible for sound stimuli, and type 2, which are thought to be more enhancers of sound. Two papers were published in the same issue of Cell that identifies 3 new molecularly distinct subtypes of type 1 SGNs, 1A, 1B, and 1C, and this was all identified by single-cell RNA sequencing, and they show that type 1C in particular is vulnerable to age-related hearing loss. The first study comes from Johns Hopkins and Decibel Therapeutics, where again the authors performed single-cell analyses of the type 1 spiral ganglion neurons, and they identified the 3 subtypes, 1A, 1B, and 1C. Using RNAscope, they validated the expression of the type 1C marker, LYPD1, in a subset of SGNs, as well as confirmed expression of COO4F1 and COB2 in differing SGN populations, demonstrating that the populations do not overlap. Furthermore, in the second study coming out of Harvard, they used RNAscope to first demonstrate that type 1 and type 2 SGNs do truly exhibit molecular differences, with the type 2 marker TH expressed highest in cells with low expression of the type 1 marker and vice versa. In addition, single-cell RNA sequencing identified a novel type 1 marker, TSC22D3, and RNAscope could confirm that TSC22D3 was expressed in cells that were negative for the type 2 marker TH, and that it did overlap with expression of the type 1 marker, EPHA4. So this confirms that this newly identified marker, TSC22D3, is a reliable marker of type 1 SGN. Furthermore, these authors quantified their RNAscope results and show that those RNAscope results correlate nicely with the single-cell RNA sequencing results, confirming the inverse relationship between type 1 and type 2 markers. In the same studies, the single-cell RNA sequencing analysis revealed 3 types of type 1 SGNs, and RNAscope was used to validate these gene expression profiles in the tissue context. They demonstrated opposing gradients of gene expression patterns depending on which subtype those genes were expressed. For instance, COB2, a gene found to be expressed in type 1A SGNs, was expressed in an opposing gradient from LYPD1, which was found to be expressed in type 1C SGNs. And again, quantification of these RNAscope results correlated with the single-cell RNA sequencing results. Finally, these researchers showed that the type 1C SGNs are particularly vulnerable to age-related hearing loss. In aging animals, SGNs are progressively lost, but in particular, SGN loss was characterized by gradual loss of cells expressing low POB2, high LYPD1, as demonstrated by RNAscope in these images. That gene expression profile is indicative of type 1C SGNs. So we were seeing a reduction in type 1C SGNs, showing that they're selectively vulnerable to age-related hearing loss. Also last year in the same issue of Nature, 2 studies profiling the single-cell transcriptome of the mouse and human airway epithelium both serendipitously identified a new cell type that could be linked to cystic fibrosis. While generating cell atlases of the mouse and human lungs, these authors also identified a new airway epithelial cell type that expresses both FOXI1 and CFTR, a gene that is mutated in cystic fibrosis patients and responsible for transporting chloride ions out of the cell. They term these FOXI1 CFTR dual positive cells pulmonary ionocytes because they seem to be involved in transporting ions across lung tissue. And molecularly characterizing this new cell type could aid in identifying the appropriate cellular target for cystic fibrosis gene therapy. In the first study from the Broad and MGH, the authors generated a single-cell atlas of mouse tracheal epithelium in which they identified functional variations in club cells based on their location, a distinct cell type in high turnover squamous epithelial structures that they term hillocks, and disease-relevant subsets of tuft and goblet cells. They also discovered the rare cell type, which I just described, the FOXI1 positive pulmonary ionocyte. That's also the major source of CFTR in both mouse and human airway epithelium. They then went on to show that knockout of FOXI1 in mouse ionocytes causes loss of CFTR expression and disrupts airway fluid and mucus physiology, phenotypes that are characteristic of cystic fibrosis. In the second study from researchers at Novartis and Harvard, they also found the same cell type, this pulmonary ionocyte that co-expresses FOXI1 and CFTR. RNAscope was used to validate that both mouse and human ionocytes co-express these 2 genes. In addition, the assay demonstrated that CFTR was highly enriched in FOXI1 positive cells, but not FOXJ1 positive cells. The transcriptomic profiling of complex tissues by single nucleus RNA sequencing has also been gaining traction. Alzheimer's disease is an age-related neurodegenerative brain disorder of which the molecular and cellular complexity remains poorly understood. MIT researchers analyzed over 80,000 single nucleus transcriptomes from the prefrontal cortex of 48 individuals with varying degrees of AD pathology, providing a first blueprint for interrogating the molecular underpinnings and cellular basis of Alzheimer's disease. The RNAscope duplex assay confirmed cell type-specific downregulation of differentially expressed genes, revealing significantly fewer excitatory neurons, which were marked by SLC17A7, with detectable NTNG1 expression in brain sections from AD patients, compared to patients that had no AD pathology. In multiple sclerosis, demyelination of the central nervous system leads to neurodegeneration, but the severity of MS between patients is highly variable, and the disability does not correlate well with the extent of demyelination, suggesting that other factors contribute to this variability. One such factor could be oligodendrocyte heterogeneity, as oligodendrocytes myelinate axons. However, the extent of human oligodendrocyte heterogeneity and its possible contribution to MS pathology remains unknown. The authors of this paper utilized single nuclei RNA sequencing from the white matter of postmortem human brains to reveal altered oligodendroglial heterogeneity in MS patients. This heterogeneity may be important to understanding disease progression and developing therapeutic approaches. The BaseScope duplex assay complemented single nuclei RNA sequencing data, and it revealed a correlation between the sequencing data and BaseScope-ish for subcluster marker expression in the white matter of human-controlled brain. BaseScope also showed by segregating pairs of subcluster markers that less than 10% of oligodendrocytes contained both RNA markers, confirming the presence of these distinct subclusters. The authors also identified, through single nuclei RNA sequencing, this new cell population, immune oligodendroglial cells. This is a population that seemed to express both canonical oligodendrocyte genes as well as genes associated with microglia, hence why they were termed oligodendroglia. Interestingly, these immune oligodendroglia are enriched in the human MS brain. Combining BaseScope-ish assay with IHC on the same section revealed expression of the microglia marker CD74 in OLIG1-2 positive oligodendrocytes in an MS lesion, confirming the presence of this hybrid cell type in the human MS brain. And while the brain and many organs have been analyzed by single-cell techniques in numerous studies, the highly heterogeneous tumor and its microenvironment is quickly becoming a top tissue to examine at the single-cell level. Researchers at the Broad Institute dissected the cell types in IDH mutant gliomas by combining single-cell sequencing profiles of over 14,000 cells from patients that had glioma with another 165 glioma patients with their profiles analyzed by bulk RNA sequencing. They found an increase in macrophage over microglia expression programs in the tumor microenvironment as the tumor grade progresses. RNAscope confirmed these results and demonstrated that intertumor variability in the microglia macrophage state correlated with tumor grade, showing that cells from higher grade tumors were preferentially associated with a macrophage-like expression state. So for instance here, the tumor MGH56 on the bottom left is a lower grade tumor and has few microglia and no macrophages, whereas tumor MGH42 on the far right is a grade 4 glioma and contains only macrophages. However, in one particular tumor, MGH43, which was an intermediate grade 3 tumor, RNAscope revealed that this tumor contains cells that co-express the microglia marker CX3CR1 and also express the macrophage marker CD163, as indicated by the black arrows in those images. So by shedding light on the cellular composition of IDH mutant gliomas, these data could aid in the design of immunotherapies targeting gliomas. Now, cancer-associated fibroblasts, or also known as CAFs, are a major constituent of the tumor microenvironment in many cancers. However, morphology remains really the most common way to distinguish CAFs, as molecular markers are neither all-encompassing nor specific. This suggests that CAFs may be a diverse group of cells made up of several subtypes. These authors utilized single-cell RNA sequencing to delineate the heterogeneity of 768 CAFs isolated from a genetically engineered mouse model of breast cancer. And they defined 4 transcriptionally diverse subpopulations of CAFs. There's VCAFs, which are the vascular gene signature, MCAFs, which are the extracellular matrix gene signature, CCAFs, which are cell cycle gene signature, and DCAFs, which are developmental gene signature. Each of these subpopulations was discriminated by different gene expression programs representing different functionality and unique spatial location within the tumor, as identified by the RNAscope assay here. For example, on the top right, using RNAscope for the VCAF mRNA markers Notch3 and KCNJ8 in combination with immunofluorescence for the vascular protein marker NID2 revealed that vascular CAFs are localized predominantly near the vasculature, attesting to their function. Furthermore, CAF subpopulations are distinct, as shown on the bottom right, with no overlap in signal between the extracellular matrix CAF markers and vascular CAF markers. This study reveals a previously unappreciated functional diversity within the tumor microenvironment that can aid in further development of tools for precision medicine. Translating potential gene biomarkers from the Cancer Genome Atlas, or also known as the TCGA, these studies have, for clinical utility, has typically relied really on PCR-based technology or IHC. However, these techniques are limited by tumor sample heterogeneity and the lack of correlation between mRNA transcript abundance and corresponding protein levels. The authors of this paper used RNAscope in a colorectal cancer cohort to measure intratumoral gene expression of potential prognostic markers that were identified by the TCGA. They found consistent results with the TCGA, with reduced GFI1 expression associated with high-grade and left-sided tumors, and reduced TNFR-SF11A expression, which was associated with metastasis and high nodal involvement. They also found cell type-specific expression, where GFI1 and TNFR-SF11A mRNA expression levels were significantly higher in carcinoma cells compared to other cell types. Now, complex tissue and cellular heterogeneity can also arise from splice variants. Through alternative splicing, which can lead to a variety of combinations of exon inclusion or exclusion, a single gene can now encode multiple proteins to increase diversity and cellular heterogeneity. However, detection of splice variants in the tissue context has been limited due to lack of highly specific tools. Therefore, we took advantage of the BaseScope assay to detect splice variants with morphological context. EGFRv3 is a splice variant of the EGFR gene that arises from gain-of-function mutation causing an in-frame genomic deletion of exons 2 to 7, and it leads to constitutively active oncogenic signaling. It's also one of the most frequent mutations found in glioblastoma patients. Now, detection of EGFRv3 in FFPE clinical specimens by IHC and PCR approaches have been developed but have been suboptimal due to the lack of specificity and spatial resolution, respectively. Therefore, a robust assay to identify EGFRv3 status within the tumor context is highly desirable. We use the BaseScope assay to identify EGFRv3 status. We first have one ZZ pair probes that were designed to span the exon junctions in order to detect either wild-type, mutant, or both transcripts. The wild-type EGFR probes span the junctions of either exons 1 and 2 or exons 7 and 8. While the EGFRv3 specific probe spans the junction of exons 1 and 8. A common probe that spans the junction of exons 8 and 9 was also used to detect total EGFR, or both wild-type and EGFRv3 transcripts. In this slide here, we show two glioblastoma cases that were stained with the probes I just described. With both wild-type probes, we see staining in both cases, indicating both of these tumors express wild-type EGFR, and that can be seen in the left column for each tumor type. However, with the mutant probe, we only see signal in the case on the left, indicating that this glioma is positive for the EGFRv3 variant, while the glioma on the right is not. So taken together, we can identify splice variant status for EGFRv3 in situ in FFPE glioblastoma samples. And researchers from Italy just published this paper looking at the HER2 splice variant, HER2 delta 16. This splice variant is characterized by the deletion of exon 16 from the HER2 gene and is associated with HER2 positive breast cancer aggressiveness, stemness, and drug susceptibility. However, with the exception of RT-PCR, easily reproducible assays are still lacking to clinically detect and quantify the HER2 delta 16 splice variant and its expression level in the tissue context. So the authors used BaseScope-ish assay to discriminate this variant from the wild-type isoform. So as you can see here, the HER2 delta 16 probe spans the junctions of exons 15 and 17. The wild-type probe spans the junctions of exons 15 and 16. And then they also have a common probe that spans the junctions of exons 17 and 18. And that common probe that common exon junction will be present in both transcripts. And they then applied the BaseScope assay to now detect HER2 delta 16 in a variety of cancer types, including breast, gastric cancer, and colorectal cancers, which we can see here. The last research area I'd like to highlight is single-cell spatial analysis for stem cells and development. So during homeostasis, LGR5 positive intestinal crypt cells drive turnover of the intestinal epithelium. However, even though LGR5 positive cells are lost during injury, the epithelium can still recover. Therefore, an additional population of quiescent stem cells must exist to drive epithelial renewal in response to injury. Researchers from Alex Grigorev's lab at Mount Sinai Hospital in Toronto used single-cell RNA sequencing to profile the regenerating mouse intestine and identify a novel stem cell population, which they termed revival stem cells, and is shown here as cluster SSC2 on the Tisney plot. They found these stem cells were enriched for genes involved in cell survival, such as clusterin, also known as CLU. During homeostasis, CLU-expressing cells are rare and are distinct from LGR5 positive cells. And in response to irradiation, the number of CLU-positive epithelial cells expands. The RNAscope Multiplex assay revealed that these rare CLU-positive cells in the normal intestinal crypt are distinct from LGR5 positive cells, and that there is a significant increase in CLU-positive cells in the damaged intestinal crypt, as shown on the right side. Furthermore, the researchers identified YAP1, a transcriptional regulator, as important for regeneration of the intestinal epithelium. During intestinal regeneration after injury, YAP1 induces reversal stem cells that will create new LGR5 positive cells and thereby restore the epithelium. Single-cell RNA sequencing revealed that the YAP1 gene signature was present exclusively in the SSC2 cluster. And deletion of YAP1 in the intestinal epithelium suppressed emergence of these endogenous CLU-positive cells, as demonstrated in the middle section by dual RNAscope-IF assay. If you'd like to learn more about this great story, please watch the GEN webinar tomorrow with Dr. Alex Grigor. Now, organogenesis requires the complex interactions of multiple cell lineages that coordinate their expansion, differentiation, and maturation over time. Pancreatic organogenesis is a very complex and dynamic process that ultimately results in the generation of multiple cell lineages that perform the functions of the mature pancreas, including regulation of glucose homeostasis by the endocrine compartment and production of digestive enzymes by the exocrine compartment. Researchers from Julie Snaden's lab at UCSF profiled the cell types within the epithelial and mesenchymal compartments of the murine pancreas across development, using a combination of single-cell RNA sequencing, immunofluorescence, RNAscope in situ hybridization, and genetic lineage tracing. Among several major findings, they also identified a previously underappreciated cellular heterogeneity of the developing mesenchyme, as shown here in the embryonic day 14.5 pancreas, and that was confirmed by dual RNAscope-IF assay. This work establishes a roadmap of pancreatic development and demonstrates the broad utility of this approach for understanding lineage dynamics in developing organs. Now, as I mentioned earlier in our talk, we have an assay that will be coming out soon, the RNAscope High-plex assay. To fully characterize and spatially map the diverse cell types identified by single-cell transcriptomics, RNA-based in situ assays capable of higher multiplexing are critical. Coming soon, ACD will release its RNAscope High-plex assay, which now allows researchers to visualize up to 12 targets on the same tissue section at single-cell resolution. With the multiplexing capabilities of this assay, researchers can now find the population of cells by using a cell-specific marker. For instance, here we're showing DRD1, a marker for medium spiny neurons in the mouse striatum. You can confirm your single-cell RNA sequencing results by simultaneous detection of additional gene expression targets, such as DRD1 positive cells that express FOXP1 and MEIS2, as visualized here in the mouse striatum. And you can really interrogate and characterize complex heterogeneous tissues by now mapping up to 12 targets simultaneously in the tissue context and identifying multiple cell subtypes, such as here we show the subtypes of DRD1A and DRD2 in the mouse striatum. To learn more about the RNAscope High-plex assay, you can register to watch the new product introduction webinar, which will be held on July 23rd. Registration links will be opening on July 10th, and you can check out more on our website. And shown here is a nice example, the compiled image from a study we performed at ACD that confirmed and spatially mapped the diverse striatal medium spiny neurons that were identified by single-cell RNA sequencing in the mouse brain from a 2016 paper. Using our 12-plex high-plex assay and the image registration software that we provide, we could visualize the heterogeneity of these cell populations on the same tissue section. So in summary, what we talked about today is the RNAscope assay and how it has a unique probe design strategy that can dramatically improve signal-to-noise ratio. We currently have more than 15,000 probes in our catalog. A new probe can easily be designed and made within 2 weeks for any target in any species. RNAscope provides both quantitative and spatial information on your targets in the complex tissue environment at single-cell resolution. Our newest assay, BaseScope, can detect splicing variants and short sequences down to 50 nucleotides, as well as validated point mutations. And RNAscope assays can be complemented by IHC or IF for simultaneous RNA and protein detection on the same slide. The growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then there have been over 2,200 peer-reviewed papers published using the RNAscope technology in numerous journals, including many top-tier ones. And the RNAscope technology is highly relevant across multiple fields of research, with over 40% of the publications in cancer research, followed by neuroscience and infectious diseases. Furthermore, the incorporation of the RNAscope technology into single-cell RNA sequencing workflows is demonstrated through the rapid growth of publications using the assay to confirm and spatially map single-cell RNA sequencing data in the tissue context, as shown here in the graph. And I would say over 80% of these single-cell papers have been published in very high-impact journals, including Nature, Cell, and Science. And so with that, I would like to thank you for your attention as we talked about incorporating spatial mapping into single-cell RNA sequencing workflows. If you would like more information on the RNAscope technology, please visit us on the web at acdbio.com, or you can contact us at the email addresses shown here on the screen. In addition, I recommend checking out some more great webinars that are coming up related to single-cell biology and spatial profiling. Again, tomorrow we'll have Dr. Alex Gregorius showing how he has used the technology to map his single-cell transcriptomes that are drivers of intestinal regeneration. On July 23rd, we'll have a High-plex new product introduction webinar to introduce you to the High-plex assay with more detail. And also on July 23rd, a little bit later in the day, we'll have a webinar co-hosted between ACD and NanoString to show how you can incorporate the two assays for molecularly guided digital spatial profiling. And so now I'd like to invite you to ask any questions you might have. Thank you very much for your time.